Hello, my dear friends. My name is Akanksha Ahire. I am working as assistant professor in the Department of Biotechnology, KTHM College, Nasik. Today, we are discussing the course that is Food Biotechnology, which comes under the STRIDE scheme. The topic of today's is detection of adulterants which are present in the oils and fats. So, let's start with our topic. In the last lecture, we have studied about the identification of adulterants which are present in the wheat and maida. In today's lecture, we will discuss which are the adulterants which are present in the oils and the fats. Now, the agenda of today's session is the first point we are discussing, that is introduction to the adulteration. Exactly what is meaning of adulteration? The second point is we will study the different examples of adulterated food. And the third point is the determination of adulterants which are present in the oils and the fats. Now let's start with the adulteration. The learning objectives of this session is the first, to introduce the concept of adulteration, to know the reasons behind the adulteration, why we are doing the adulteration, and the third, that is you have to just detect the adulterants which are present in oils and fats. Let's start with the adulteration. So what is the adulteration? Adulteration is nothing but it is a simple process in which what happens, something is get added or something is get substituted. Okay, now we will study the actual definition of adulteration. So what it is exactly? It is intentional addition or substitution or abstraction of substances which are adversely affect the nature, substance and equality of food, but also their incidental contamination during the period of growth, harvesting, storage, processing, transportation, and distribution. So, what is adulteration? It is a simple process in which intentionally something is get added or intentionally something is get substituted. Now, why we are doing this adulteration? There are different reasons behind this adulteration process. The first reason is the possibility of making greater profit. The second reason is masking of food spoilage and ignorance of the people handling food. And the third reason behind the adulteration is increasing the bulk or the quantity of food atom by adding a cheaper substitute in the most common ways of increasing the profit margin. So why we are doing this adulteration process, or why the peoples are doing this adulteration process? The main three reasons are there. The first very important reason, that is for the greater profit, to avoid a food spoilage and for the profit margin. Now you can see there are different examples of adulterated foods are there. Different foods are present in the market which are get adulterated. So you can see the first example, that is the adding of water to the milk or the stone to the food grain. You know that in a milk water is present. It means water is the adulterant. Okay, and because of presence of water in a milk, that milk is get adulterated. Okay, the first example of adulterated food in the market, that is the milk. And again you can take a second example, that is a food grain. The next is the sweet meat maker may place aluminium foil instead of silver foil on the sweets he makes. This is a second example of adulterated food. Next, there are some perishable foods are there. Okay, that perishable foods, they are get spoilage very fastly. Okay, to avoid this thing, some peoples, they are just coated that spoilage food with various adulterants. The next, there are some poor quality fruits are there, some vegetables are there, some pulses are there. Okay, they are sometimes artificially colored to give them for the fresh look. Okay, so there are some foods in the market, they are the artificial colored fruits and vegetables are also there. And the last one is the adulterants like artificial colors are get added to the food to improve their consumer appeal. For example, they are giving a rice and meat preparation or the sweets. So you know that in a biryani or in a different sweets, different artificial colors are get added. So you can see these all are the different examples of adulterated food which are present in the market. Now you can see here, these are some pictures of adulterated food, that is sugar is there, jam is there, some milk products are there, cereals are there, rice, wheat, coffee, oil and fats, honey, spices. These all are the foods in which adulterants, different adulterants are there. Now, exactly what is meant by adulterants? Adulteration is a process in which what happened, adulterants are get added. Okay, but you should know what is meant by adulterant. So what is the adulterants? Adulterants is any substance which is used to adulterate a particular atom of a food is called as adulterant. It means simple, adulterants is called as the any substance which is get intentionally added into the normal food. It is any substance that lessen the purity of effectiveness of any substances. So what happens when you are adding a adulterant to a food atom? Then what happens? The purity of that substances get reduced. Now you can see in a tea, suppose tea is a one food atom, and in that tea the color, iron filling, foreign leaves, exhaustless leaf, these all are the adulterants, or these all are the other substances which are present in the tea. Again you can take example, suppose pulses are there. Okay, so in a pulses artificial color is there, foreign starch is there, extraneous matter is there. Okay, so what this artificial color, foreign starch, this extraneous matter, these all are the adulterants. These all are the foreign substances which are present in the pulses, and because of the presence of this adulterants, that food is get adulterate. Again you can take an example, that is cereals and their products. In cereals and in their product, some pesticide residues are there, some powder chalks are there. Okay, so these are the adulterants which are present in the cereal and their product. Now we will turn towards the detection of adulterants which are present in the oils and fats. There are different methods and there are different techniques by using you can detect which adulterants are present in oils and the fats. Before we are directly going to the different methods and technique, you should know what is the oil and the fats. So you know that there are different vegetable oils are there, there are different edible oils are there. Okay, this vegetable oils and this edible oil, they have some nutritional value. The very important component of this oil and fat is the triacyl glycerol. But nowadays what happens, some adulterants are get added in this oil and fats. These adulterants are not good for your body, or it is not good for your health. So you have to just remove that adulterants from this oil and fat. So for the removing, first very important first step is that you have to detect the adulterants which are present in this oil and fats. Now there are different types of adulteration. Okay, the first adulteration method is admixing a cold press oil with a refined one, and the second type of adulteration is replacement of more expensive oils and the fat with a cheaper one. By using this two types of by using this two method of adulteration, you can adulterate the food. The first method, that is detection of rancidity in edible oil and the ghee. Now you can see rancidity is the adulterant which is present in the edible oil and the ghee. Now you have to check the rancidity. Okay, that is you have to check the adulterant which is present in the oil and ghee. Now for the detection of rancidity in the edible oil and the ghee, two reagents are very important. The first reagent is, that is 0.1% fluorogeucinol in diethyl ether solution. Now, how you can make this solution? You have to just weigh 100 mg of fluorogeucinol and dissolve that in 100 ml of diethyl ether. This is a first reagent, and the second reagent is concentrated HCl. Now, this is a procedure. What you have to do, you have to just take 3 ml of sample of oil or a melted sample of fat in a clean test tube. In that test tube, you have to add 3 ml of concentrated HCl. This is a first step which you have to do. The second step, you have to just shake that tube very carefully to mix the content. The next, you have to add 3 ml of 0.1% fluorogeucinol solution in ether. After adding this solution, you have to again shake that tube very vigorously for 2 minutes and keep that test tube as it is. After that, after half an hour, you have to just examine that test tube. Now, this is a result. So what is the result? The first result is that red or pink coloration in the acid layer, it indicates that the oil and fat sample in rancid, and the absence of color development, it indicates that oil is not rancid. It means after doing all this procedure, after doing all this step, at the last, if red color is there or if pink color is there, it means that the given sample is it in that in the given sample rancidity is there. If suppose there is no color development or the absence of color, it shows that or it indicates that there is the no rancidity is present in a given oil sample or in a given fat sample. The second method or the second test, that is a detection of Argemone oil. Okay, for this again you will require a three reagent. The first reagent is 2% salicylic acid in methanol. How you can make this? You have to just weigh 2 gram of salicylic acid and dissolve it in methanol. The second reagent you will require, that is concentrated HNO3, and the third reagent which you require, that is concentrated H2SO4. The procedure. Okay, so what you have to do, the first thing, take a 5 drops of oil, that is your sample, in a clean dry test tube. After taking a sample, you have to add three things in a sequentially manner. The first thing you have to add, that is 0.5 ml of 2% salicylic acid solution. The second thing you have to add, that is 2 ml of concentrated HNO3, and finally you have to add 2 to 4 drops of concentrated H2SO4. After adding all these things, you have to just mix this thing very carefully and observe the development of color after 20 to 30 seconds. Now this is a result. A crimson red color within 20 to 30 seconds indicates the presence of Argemone oil, and a no color change is observed, that is a pure oil. It means suppose in a given sample, after addition of all this solution, if red color is get observed within 20 to 30 seconds, then you can say that in a given sample Argemone oil is there. And if there is no color change, it confirms that that oil is the pure oil. The third method is detection of cottonseed oil. For this, again, you will require a three reagent. The first is amyl alcohol. The second is precipitate sulfur 1% wet by volume solution. How you can make this solution? You have to dissolve 1 gram of sulfur in 100 ml of carbon disulfide. And the third reagent which you require for this, that is a Halpence solution. Okay, how you can make this Halpence solution? Mix a equal volume of amyl alcohol and the sulfur solution. Now this is a procedure. So what you have to do, take a 5 ml of sample in a glass stopper test tube, then add equal volume of Halpence solution and you have to just mix that test tube on the vortex. Then heat that test tube gently in a water bath at 70 to 80 degrees Celsius with occasionally shaking till the CS2, that is a carbon disulfide, has boiled off and the foaming ceases. Next, you have to place that test tube in an oil bath and maintain at 110 to 115 degrees Celsius and hold that bath for the 2.5 hours. And after 2.5 hours, you have to just observe the test tube for the color development. Now, this is a result. A red color seen at the end of this period, it indicates that the presence of cottonseed in oil, and there is no color development, it indicates that the absence of cottonseed in the your given sample. It means in a given sample, if red color is get developed, then you can say that in a given sample cottonseed oil is present. And if there is no color development, then you can say that the given oil sample is a pure oil sample. There is a absence of cottonseed oil. The fourth method is detection of mineral oil. For this, you will require only one reagent, that is alcoholic potash. Okay. For the making of this alcoholic potash, you require or you have to do the dissolve 8.6 gram of KOH pellet in 2 ml of water and then make up the volume to the 100 ml by using aldehyde free alcohol. Now this is a procedure. Take 2 ml of oil sample and add the equal quantity of alcoholic potash. It means you have to add 2 ml of alcoholic potash. The second step you have to do, that is heat in a boiling water bath for 15 minutes. After 15 minutes, add 10 ml of hot water in that test tube and look for the turbidity. Now here you have to check the turbidity. Now this is a result. So what happened? any turbidity shows the presence of mineral oil greater than the 1%. If turbidity is there, it confirms that in a given oil sample, mineral oil is present more than 1%. The depth of turbidity depends upon the percentage of mineral oil present. It means how much that sample is turbid. Okay, or how much turbidity is there, it is depending upon the how much amount or how much percent of mineral oil is present in your given sample. And clear solution indicates that the absence of mineral sample. Okay, it means turbidity is there, in it means in that sample or in that given oil sample the mineral oil is added, or if clear solution is there, it indicates that the mineral oil is absent in a given oil sample. The next step is detection of castor oil in the edible oil. Now for that, these two reagents are very important. The first is petroleum ether and the second one is the ammonium molybdate reagent. How you can make this? You have to dissolve 1.25 gram ammonium molybdate in 100 ml of concentrated H2SO4. Now this is a procedure. Simple procedure is here. Take 1 ml of sample in a dry test tube. Add 10 ml of petroleum ether in that. Shake that test tube vigorously for 2 minutes. After 2 minutes, you have to add 1 to 2 drops of ammonium molybdate reagent. Now this is a result. Instantaneous development of turbidity indicates the presence of castor oil and the transparent solution indicates the absence of castor oil. Again you have to check the turbidity here. If turbidity is there in a given oil sample, it confirms that in a given oil sample castor oil is get added. It means castor oil is the adulterant which is present in the given oil sample, and if sample is transparent then it indicates that the castor oil is not get added into your given sample. The next test or the next method is the detection of adulteration in a coconut oil. Now for this, no reagent is required. You have to just do directly procedure. The first thing you have to do that place a small bottle of oil in a refrigerator at a temperature 5 to 10 degree Celsius, and you have to check that bottle after 60 to 90 minutes. Then what is the result after 60 to 90 minutes? A pure coconut oil solidify completely with no separated layer. If adulterate, separate layer. If adulterant oil is visible, it means in a coconut oil, suppose adulterants are there, then what happen? Separate layers are get formed. But if coconut oil is pure, then there is no separate layers are get formed. Now, so what is the summary of today's session? What we studied in a today's session? The first thing, that is what is meant by adulteration or what is meant by adulterant. The second thing, we learn about the different examples of adulterants and adulterated foods which are present in the market. The third thing, we studied that what are the different reasons behind this adulteration. And the fourth very important point we studied today, that is the different methods which are used to detect the adulteration which are present in the oil and the fats. Thank you.